AGAROSE Gel Electrophoresis, a step-by-step guide. This technique is based on the separation of nucleic acids based on their sizes, under the influence of electrical charge. Agarose molecules form polymers with big intermolecular spaces, through which DNA or RNA fragments migrate. Nucleic acids being negatively charged, will always migrate towards the anode, through the minute pores of the agarose gel. At first we shall see how to prepare the gel. Measure and weigh agarose powder. Add agarose powder to a flask with appropriate amount of buffer solution. Heat and stir the mixture in a flask to dissolve the agarose till it forms a homogeneous solution. Pour the gel into a gel caster and insert the gel comb into the liquid gel. Take the comb out as the gel solidifies, and thus it will leave impressions, or what we call sample loading wells, where the samples would be loaded. Mix nucleic acid samples with loading dye, which also has ethidium bromide. Ethidium bromide gets intercalated between the gaps of nucleotides and when subject to UV light, will fluoresce, thus showing the respective positions of nucleic acid fragments on the gel. The percentage of agarose used depends on the size of fragments to be resolved. If the samples are small fragments around 500 base pairs, then around 2% gel is used to get better resolution of bands. Bigger the fragment range, lesser is the percentage of the gel. Here we can see that the nucleic acid samples have been loaded into the wells. A blue-colored dye such as bromophenyl blue is also added to the sample to track the migration of samples through the gel. Since DNA is not visible under normal light, we have to track the movement based on this tracking dye's movement only. Place the gel in the electrophoresis chamber, fill the chamber with same buffer solution and turn on the electricity. As the blue tracking dye almost reaches the opposite end of the gel, now it's time to visualize the nucleic acid bands. Place the gel in a UV transilluminator machine to see the bands, which corresponds to fragment sizes of nucleic acids. The smaller fragments of nucleic acids moved more conveniently towards the anode. So, what are the uses of agarose gel electrophoresis? To separate DNA or RNA. To estimate size of nucleic acids. To analyze PCR products, e.g. in molecular genetic diagnosis or genetic fingerprinting, and many more. Estimate the size of DNA fragments with reference to previously known size markers. Thank you for joining us in the lab today. If you found this video helpful, please like and subscribe for more educational content. Feel free to ask any questions in the comments below, and stay curious.